So to be fair,
Okay. So thus far, we've been talking about how we eat food, right? So the parts of our GI tract, the different cell types lining the GI tract. We've talked about what's going on in the different organs. We've talked about how we digest our different types of macromolecules and what's going on in terms of the different enzymes we're using for our carbs, our proteins, and our lipids. Last time we ended by mentioning a little bit about vitamin absorption and what's going on with our solutes. So like what's going on with our sodium, our chloride, our potassium. Last slide of the set was talking about the absorption of water. So we do want to know that if you drink water, right, it ends up in your GI tract just like food does, plus your food has water in it. And we've been adding in water as we add in all these gastric juices, the pancreatic juice, right? Like that chyme really is kind of smoothie texture. So we need to reabsorb that water that we've been using. So most of this happens before you get to the colon, although when we come back to the colon, after a brief detour talking about the secretion of enzymes, we'll see that there's a little bit more going on with the water at that point. But for what we know so far, the secretions that we're about to talk about in this next section add up to about like 7 liters of water per day. So you don't want to be losing that much water from your system, right? Like you want it back in your plasma. You want to keep blood volume up. You only drink about 2 liters of water per day. So the math doesn't math if you were to excrete all those 7 liters. So we gotta take them back. Fortunately, that absorption process is passive. So like in a lot of other places, basically we're building up a solute gradient. So as we move, say, sodium, right, as we move that out of the lumen of the GI tract into the neighboring cells and across, especially across, using our pumps on the basolateral membrane, the water is going to follow across along with those solutes and kind of be dragged. So water is just really getting absorbed by osmosis for the most part. Okay. So recommend taking a look at these checkpoint questions that are also at the end of this section about absorption of your macromolecules in your book. So you want to make sure that you can tell how are we digesting each of these different types of molecules, our carbs, our proteins, and our fats. We want to know what can actually make it into a cell. So our carbs, we got to get those monosaccharides. Proteins, they're big, so we do have to do a lot of breaking them up, and we had a number of different kinds of enzymes, but we could maybe pull in a dipeptide, a tripeptide before we break it down inside the cells. We talked about the fats and the lipids and how we need to emulsify them because they're hydrophobic. So make sure you can kind of walk yourself through those processes at this point. Okay. So we're going to move into our next section, thinking a little bit deeper about how we control digestion, right? We don't think about what our gut is doing unless our stomach is rumbling, but your brain does actually control what are we secreting, when are we secreting it, why are we secreting it. So we're going to see that there's integration, particularly with our endocrine system, but also with our autonomic nervous system and these various organs that we've been talking about. And then after we talk about that for a bit, we're going to come back and think about movement, so motility of the gut and how we're actually squeezing stuff along. Okay. So we're going to start out by thinking about some reflex pathways and some hormones that are going to let you effectively digest your food. Okay. So confusing terminology a little bit. Specifically, I don't like that they call them phases. But we have three kind of types of gastrointestinal control. And you might kind of think about them as phases if you're thinking about like you ate a burger, swallowed it. We're going to step through in order. We're going to have some signals coming from your head. So anytime you see that CEPH beginning, Seth, that has to do with the head. That's like a Greek root for head. Gastric phase, we're going to have some reactions that happen when we get to the stomach, right? So like we have gastric juice, gastric is the stomach. So gastric phase signals are going to come from the stomach. And then as we pass out the pyloric sphincter into the duodenum and the small intestine, we're going to see that we have some additional kind of fiddling with the levels of our various GI hormones and secretions by signals that are being sent by the intestines themselves. So if we're thinking about like what these might be, for the stimuli originating in the head in the cephalic phase, like by head, we really kind of mean brain. So literally digestion and the digestive process kind of starts when you see food, when you smell food, when you use those sensory organs in the head, right? So you walk by Einstein's and you smell some bagels or some pastries and your mouth starts to water. That's because of this cephalic phase control. Okay, so this can just be even just thinking about food can start out some of these digestive processes, right? So seeing an ad and suddenly wanting whatever it is they're selling, right? Like they're taking control over this cephalic phase, kind of gastrointestinal control. Okay. Now, in order for this to work, we are going to need our nervous system, and we're going to need some pretty long nerves, right? Because your head, your stomach are not particularly close to each other. So we're going to have some long reflexes involved in the cephalic phase of GI control. So we can tell, and we're going to term them long reflexes when we have this input from the central nervous system, right? We're going to have to go like a longer kind of pathway. Okay. When we get to the gastric phase, so saw the burger, you smelled the burger, you swallowed the burger, traveled through your mouth, your pharynx, your esophagus, it's down in the stomach. That's time for our gastric phase control. Now we're going to have some long reflexes again, but we're also going to have some shorter reflexes. So we're going to have some shorter pathways involved here, as well as our GI hormones getting involved. So we're linking into the endocrine system. We're linking into those hormones that travel around in the blood. Same thing with the intestinal phase. So once we are starting to get signals from our small intestine, we're going to have long reflexes, we're going to have short reflexes, and we're going to be thinking about those hormones. So all along this way, your body is trying to tell you about, do you have food in you right now? How much, what kind, and what should we do about it? So what we're controlling when we're talking about GI control is we're really controlling what your body's making in order to break up that food. So we're going to be controlling the contents of the saliva, right? So we know that our digestion of carbohydrates can start in the mouth with that salivary amylase. And we know we need that saliva to kind of lubricate food. Then we're going to be controlling what's going on in the stomach, the production of stomach acid, and that activation of pepsinogen to pepsin that's linked to that, right? So we're going to need to send signals to those chief cells and the parietal cells. We're going to have to tell the pancreas to make enzymes, to make bicarbonate. We're going to have to tell the liver and the gallbladder that we need some bile so that we can emulsify our fats. We're doing this with all these different reflexes, hormones, all this signaling stuff. Okay. So we're going to start with the saliva, right? So we're going to work the way from our head down. Okay. So when we have this cephalic phase, of gastrointestinal control. We can even include taste in there, right? Your tongue's in your head, so we'll put it in there. So when we have food in our mouth, we can taste it, we can feel that texture. So we're going to have receptors in the mouth that can sense these things. So mechanoreceptors, think mechanical, that's going to be like the texture of the food stuff getting perceived. Chemoreceptors are going to be things in like your taste buds, sensing what chemicals you have, like acidity is sour because you have receptors sensing those protons, those hydrogen ions. We have more complicated receptors in the mouth that we won't go into, but we can sense what we're eating, right? Okay. So what's going to happen at that point, right? You can taste your food, you can feel your food. We're going to send a signal up to the medulla oblongata. We have a salivary center in that part of the brainstem, basically. Okay. So in the medulla, we've got the salivary center that's going to send out signals to the autonomic nervous system. So remember, autonomic nervous system, it's not really under your conscious control, but it's important for homeostasis. It has those two branches, the fight or flight part of your nervous system, that sympathetic part, and the parasympathetic part, that rest and digest portion, right? So here's where we get that digest part in. Okay, so we're sending a signal up to our brain, up to the medulla, telling us we literally have food in our mouth right now. That medulla oblongata is sending back signals to your body, telling you what to produce. This is also where we'd be processing seeing food, smelling food. It would be going to that same kind of salivary center, controlling how much saliva you're producing. Okay. So the parasympathetic part of the nervous system is the digest portion. When we have that parasympathetic activation, you can feel, like literally feel, that you are producing more saliva. The saliva is going to be kind of watery. We're going to be adding water in there. We're thinking that kind of like lubrication piece, that swallowing piece mixing around with the food. Sympathetic activation, though, does also affect this process. So when we have sympathetic activation, so that fight or flight portion, you might feel like dry mouth when you're stressed out, even if you have enough saliva, right? Like you can even feel like your mouth is dry, even though there's spit in there. And this is because with sympathetic activation, the contents of the saliva is actually different. So the contents of the saliva with sympathetic activation has more like proteins in it. It's less watery and more viscous. So even though you're like producing saliva with sympathetic activity, your tongue can kind of sense that it's not this same quality as we would have when we're just kind of like resting. So that's in the mouth. All right. Now we are going to be moving down to our stomach, really. Okay. So remember our stomach, we've got a bunch of things that we're making, but from the digestive process portion, we were particularly interested in the pepsinogen and the hydrochloric acid that we're making. That gastric juice is really acidic. We know we have that pH of 2 that allows us to denature the proteins, unfold them, right? Also can act as part of the immune system. Lots of stuff can't survive a pH of 2. That's really low. So we're going to want to control that. And we're going to want to make sure that's matched to our nutritional state, right? Like matched to when we're eating food, right? So we're going to be sending signals to the stomach about acid secretion as well. So we're going to see that acid secretion is under the control of the parasympathetic nervous system. It's also going to be linked up with the secretion of gastrin, which is that hormone coming from the stomach, basically. So we're going to have some kind of like, you can almost think of it as feedback there. And histamines, right? So that kind of inflammatory signal that we think about in other parts of the body also influence acid secretion as well. Okay. So our acids, making basically protons, hydrogen ions, right? Their job with respect to the molecules we're digesting, food we're digesting, is really to unfold them, right? But really to activate the pepsinogen. So pepsinogen was that zymogen, right? So that inactivated form of our enzyme that starts this process of breaking down proteins, right? So the only way we break proteins in our mouth is like with our teeth, right? So physically kind of separating apart strands of muscle fiber or something. But we don't start to chemically digest proteins until we hit the stomach. So that acidic environment of the stomach, that pH of 2, changes the shape of pepsinogen as it's secreted into the stomach, turning it into pepsin that can break down the food. Okay. So we got to link up our pepsinogen secretion with our acid secretion because they're working together, right? There's not a ton of a point of secreting a bunch of pepsinogen if you don't have a bunch of acid to activate it. So we're going to see that those are linked together. So we see the same kind of signals for the secretion of pepsinogen as we do for acid. So we're going to see activation of the parasympathetic nervous system. We're going to see our hormone gastrin and some effects, some like local effects from histamine potentially. Right. So in terms of what's going on to make these things happen, right? So from the cephalic phase, so from our signals from the head for the secretion of the acid and the pepsinogen, we're thinking, we saw food, we tasted food, we smelled the food, we chewed the food, even swallowing the food, right? You can physically feel swallowing, right? So all of this is sending signals up to the medulla oblongata, right? And what that is doing then is going to send out signals through the parasympathetic rest and digest branch of our autonomic nervous system. And this is what's going to be stimulating secretion, basically. So we're activating our parasympathetics. The parasympathetics, we've got like a domino effect. They activate our hormone gastrin, right? And in total, we want to increase how much acid and pepsinogen we have, because before, if you got an empty stomach, what are the acid and pepsinogen going to do, right? We have a mucus barrier with some bicarbonate protecting the stomach wall, but really the only protein in an empty stomach for them to digest is like the tissue of your stomach. So we don't need that much acid and pepsinogen until we eat stuff, but we do want a bunch of acid and pepsinogen as we are eating, right? In case you eat a big protein heavy meal. Okay, so here's what that pathway would look like. So remember, green is telling us what our stimuli are. This purple blue is telling us about the response in our body. The red orange is telling us about the result. So this is showing us what's happening with the cephalic phase, specifically control of the secretions from your stomach. So our stomach acid, pepsinogen, our gastrin. So we are sensing the food with our face, our organs, our sensory organs. This is how we're defining the cephalic phase of control, right? This is activating the parasympathetic part of our autonomic nervous system. So here we see that increase in parasympathetic activity. So if we were talking about the mouth, this would affect the saliva secretion, right? We'd start to produce more saliva in order to lubricate that food, maybe break down carbohydrates. When we're thinking about the stomach, we're getting ready to break down any proteins in the food. So we are going to directly tell the parietal and chief cells in the gastric pits, in those walls of the stomach, to make acid and pepsinogen, right? And we've got a little side loop with extra like double signaling, right? So we've got hormones coming from the G cells, which are also in the pits of the stomach. what we're doing is secreting gastrin. So gastrin would then get into your plasma, get into your bloodstream, loop around, and that gastrin would also be doubling back to its neighboring cells as that plasma travels around, telling them to make more acid and pepsinogen. This is useful. We might be eating protein, so we're making more of these secretions that let us break down protein. And we've got a hormone that's telling the cells that need to make those secretions to make secretions, and also might be telling other parts of our body about these signals, about this input that we have right now. Okay. Now, that was all from the cephalic phase, right? So all the signals for that part of the control were coming from, did you see it? Did you smell it? Did you taste it? Okay. Now, to talk about gastric phase regulation, I'm emphasizing this, right? Because like, just because the reaction is happening in the stomach doesn't mean that its control is gastric phase. So it's only gastric phase control when the signal is coming from the stomach. Okay. So cephalic phase told us to make all these secretions coming out of our gastric pits. Now, once we have that food physically in the stomach, we're digesting it, right? And in particular, we're breaking up those proteins, right? So we're gonna have receptors that can sense, right? We have stuff in our stomach, right? So we can sense that we've got proteins in the stomach, right? So proteins are big tertiary and quaternary forms, so all folded up before they denature, right? They get denatured, unfolded into the polypeptides that we start to break up, chop up with our pepsin, right? Which we've activated using that stomach acid. And so at that point, maybe we got some amino acids, depending how we chopped it up. Probably mostly still kind of larger chunks, but food spends a fair amount of time in the stomach, so could break it a bit farther. So those are some of the stimuli that we'd be thinking of when we think about gastric phase regulation, is like literally the presence of food in the stomach is creating a reaction. Another thing that you'd sense, other than the like chemical contents of the stomach, is you'd sense like how big your belly's getting, right? So your stomach has all those folds in it, the gastric rugae, right? The wrinkles, right? And it's unfolding like an accordion as you fill it, okay? So we're sensing stomach distension as well as that stomach fills up. Okay. So this gastric phase regulation is a reaction to literally having stuff in the stomach. We're sensing proteins and broken down protein products, as well as the stretch on those stomach walls. And you're triggering basically the same things as we did with the cephalic phase. Okay, so it's a different origin for why we're doing this, but we're having a similar effect, right? So we're again going to be triggering gastrin release. We're going to be triggering the release of acid, and we're going to be triggering the release of pepsinogen. So that food's going to spend a while in your stomach, basically. So once the food hits the stomach, right, it's not like the burger patty gets to your stomach, okay, now you're done, turn off the pepsinogen, we don't need to break it up anymore. You want to keep going. So we're going to keep needing that pepsinogen and acid for a while, and gastrin is our hormone telling us about all this stuff. So we're going to keep that signal going too. So effects similar, but origin of the signal is different here. So here's what that looks like. Okay. So what we're looking at now is that gastric phase control. Okay. So we've got almost identical figure here, right? So our differences now are that our reason for the signal is different. So we've got the proteins and broken down proteins in the stomach, as well as literally feeling that stomach distending, right? So that's our initial signal. We've got receptors that sense this stuff. So we mentioned them in the mouth, but didn't write them out. But now we've got those chemoreceptors and mechanoreceptors listed out. Okay, and our overall signal here is going to be to tell those parietal cells and the chief cells to make more acid and pepsinogen. Okay. We can have some shorter pathways for this signaling and some longer pathways for this signaling. And when we think about that, we're going to be thinking about the hormone gastrin, which we're triggering with these gastric phase stimuli, just like the cephalic phase stimuli. Gastrin, that hormone's getting into the blood. Cells in your stomach can sense it, make more acid and pepsinogen. What we don't see here, just to note, is that parasympathetic bit, right? So if we remember the branches of our autonomic nervous system, right? So autonomic nervous system, right? As we're coming to the end of the course, worth reviewing as we go along now that we can link together all sorts of topics, right? We know that we have the parasympathetics and we have the sympathetics. And we have all sorts of differences between them. But one of them was where these nerves are coming from, right? So our parasympathetic innervation is all craniosacral in origin, right? So parasympathetic signals have to come from your cranial nerves or like from your pelvis, right? Nothing in between, right? So if the signal is coming from your stomach, it's not coming from a cranial nerve, right? So that's why we don't see the parasympathetic reaction here on this gastric phase of control, is because those parasympathetic nerves, they're starting in your head. So if they're sending out a signal, we got to put the head and the brain kind of in that map. Okay. So this is really more linked up with these kind of hormones is what we're really emphasizing here. Okay. Cool. All right. So in addition to telling us to make more acid and pepsinogen in order to break down proteins, bless you, some of the time we're going to want to tell the stomach to stop, right? So we are also going to have cases where we want to inhibit the secretion of the pepsinogen and the acid. Okay. So from the gastric phase, right? So signals coming from our stomach. You eat your burger patty, right? Eventually, after we mix all that protein up with our gastric juices, turn it into chyme. That chyme is leaving your stomach, right? We've got that pyloric sphincter letting us trickle by trickle, kind of let that chyme into the small intestine. At some point, we don't have food in the stomach anymore, right? So that exiting of the food from the stomach, particularly that exiting of our protein products, as well as the shrinking down of the stomach, right? We might sense. Okay. So as that happens, we are going to notice now that we don't have stuff to get broken down, if we keep producing acid, we'll see that the acidity of the stomach is increasing. So all of this is going to basically tell your stomach to stop making those things we use for digestion. And that increase in acidity is in particular going to tell us to stop making gastrin. So gastrin being that hormone coming from the G cells in the gastric pits. Right? So gastrin is our hormone telling us, yes, make more of the pepsinogen and the acid. So if we turn off that gastrin signal, when we see this increasing acidity, then we don't have gastrin in the bloodstream anymore. So this is kind of a signal to your body that we don't need to be doing this digesting in the stomach anymore. Okay. So at the same time as we have the food leaving the stomach, right? It's going somewhere, it's leaving the stomach and going into the small intestine. So we're going to link up this inhibition in the gastric phase with some things that are going on in the intestinal phase of our kind of GI control and signaling. Okay, so when we get to the intestinal phase, we're going to see we have sensors. Basically, we're going to have receptors that can sense that we now have food in the duodenum instead of the stomach. So what we're going to sense then, all right, this last one, this distension is just stretch, right? So physically feeling that stretch, there's more food in the small intestine, more food in the duodenum. But we're also going to sense some of the stuff that's in that chyme coming through. So what we're going to be particularly sensing in the intestinal phase, so we're going to sense an increase in osmolarity, right? So osmolarity, right, is like our pull on water, basically. So we're going to be sensing the fact that we have all kinds of stuff coming in the chyme, right? Including maybe salt, definitely including these amino acids, all sorts of solutes, right, are affecting that increase in osmolarity. Okay. We are also going to be sensing the lipids coming through, right? So your stomach was really sensing a lot about the proteins. Your intestines, your duodenum, are sensing a lot about the fats, right? So fats, as well as that acidity coming out of the stomach, right? Because we mix the acid in with the food. That's what our chyme is made of. So as that chyme passes into the duodenum, now the environment inside the duodenum is kind of acidic, and it can tell, right? That's a signal that the stomach has passed us food. Okay. And all of these really are a signal that food has gone into the duodenum and it's no longer in the stomach. All of that is to say, right, like the effect of this is going to be, if your food's in the duodenum, you don't need to keep making stuff in the stomach to digest the food because it's not there anymore. So this is also inhibiting those gastric secretions, right? So ways we can inhibit acid and pepsinogen secretion, which are what we need in the stomach to digest proteins, are we can sense that the food left the stomach. That would turn off signals telling us to make acid and pepsinogen. Or we can sense that food is now in the duodenum. And so by process of deduction, your body knows if it's in the duodenum, it's not in the stomach anymore. So this also inhibits these stomach secretions. So these two things work together. They're just like, they're two peas in the pot. They're going hand in hand. Okay. So that's basically what we need to know about how we are controlling the stomach and its secretions, right? So we know that some of our signals come from the cephalic phase, what we see, smell, taste, right? So that gets us started making saliva, that watery parasympathetic saliva. It's also starting out with that parasympathetic activation to make acid and pepsinogen. Then as food passes into the stomach, we feel those proteins, protein products, and that swelling, still telling us to make acid and pepsinogen. Our hormone gastrin is getting in there, kind of increasing these signals, doubling us up. When food passes into the duodenum, into the intestine, we turn off this signal. We can sense it both because our stomach's kind of empty and because our duodenum's kind of full. But we have not fully digested our food yet, right? So what we need to do next, as that food is passing into the duodenum, right, we need to keep digesting that protein, but now we need those pancreatic proteases, right? So we need those enzymes coming out of the pancreas. And we're going to need the ones that can break up proteins, and we're going to need our lipases. We're going to need the ones that can break up fats. We're going to need the pancreatic amylase, so we're for breaking down carbohydrates. Right? Now that we're in the small intestine, we've got that acidic chyme coming in. We're also going to want to buffer out that acidity with a bunch of bicarbonate. We're going to need bile salts coming in from the liver and gallbladder to help us emulsify the fat. So we've still got a lot left to do. Okay, so we're not done with digesting. Now we just have to talk about what's going on with the pancreas. So we're kind of going like gland by gland, basically. Okay, so when we're thinking about the pancreas and what's in the pancreatic juice, we listed them out before when we talked about the pancreas, but now we're going to be thinking about them in a little more depth. So we got a separation between our acinar cells and our duct cells, okay? So our acinar cells coming from the acini, so these like little lobes in the exocrine pancreas, where enzymes are coming from. All right, so other things too, but the big word association bit that we want to do is associating those acinar cells with all of our digestive enzymes. So at least it's all of them, not just the proteases, not just our lipases. We've got all the enzymes coming from kind of the same place. Okay, so acinar cells are making these digestive enzymes. The enzymes, as they're coming out, being made by the acinar cells, we don't have like a huge volume, right? This is not a very like liquidy fluid when we're thinking about the secretions of the acinar cells, although we do have a little bit of water and electrolytes in there. What makes up a large volume of the pancreatic juice is what's coming out of our duct cells. So the duct cells are really making a lot of fluid with bicarbonate in it, basically. So our acinar cells are going to be more responsible for digesting the food. The duct cells are going to be more responsible for making sure you don't like burn your insides with stomach acid. That's their two jobs, basically. Okay. So we've got molecules that are controlling this process. So when we are thinking about these two pieces of the pancreatic function, right? So the two like components of the pancreatic juice, we're going to think about them like kind of separately. Okay. So we're going to be thinking about those enzymes from the acinar cells, and we're going to be thinking about the bicarbonate from the duct cells. Okay. And we're going to be linking those up with two kind of stimulants, two signaling molecules involved here. So one is CCK and the other is just called secretin. Okay, so they named that one at least pretty easily. Okay, so what the CCK does is it's a signal for the acinar cells to make more enzymes. Right, so we're kind of going to link those two things together. All right, so the primary job for that CCK is going to be to tell the acinar cells to make our proteases, our lipases, our pancreatic amylase. Okay, that's its main job. Has secondary job. So the secondary job here is it's a little bit of a stimulus for bicarbonate. And it does this kind of like domino effect where the CCK, its presence, makes the secretin like more effective. Basically, that's what we mean by potentiates. So as it's like a small stimulant for the release of bicarbonate, it's also making that secretin a little bit more effective at controlling the bicarbonate. Okay. So secretin, at least from a word association perspective, is going to be the main molecule in charge of those duct cells and their bicarbonate secretion. Okay. So that's its main job. But it's running kind of like in parallel to that CCK. So like the CCK had the secondary job of helping us with the bicarbonate release, the secretin has a secondary job. It's a little bit helping with the enzyme release, and it's doing a bit more potentiation. So potentiation is basically that it's making it more effective at letting the CCK affect the enzyme release. So technically, these are two separate processes, right? Like could control the CCK versus the secretin, but we can see that there is like a little commingling of the effect here, right? And as we have the chyme passing into the duodenum, which is where these pancreatic juices are going to go, we do kind of want both of these functions in that pancreatic juice, right? We want the bicarb and we want the enzymes. Okay. So associate CCK with acinar cells and enzymes, and the secretin with the duct cells and the bicarbonate, but know that there's like a little crisscrossing in their kind of secondary functions and jobs. but we have different triggers for the release of each of these. Okay. So the CCK is the one affecting the acinar cells, telling us to make a bunch of enzymes. Okay. All right. So it's digesting food actively. So the signal for the release of CCK, right, you want to think about this as kind of intestinal phase control, because what's happening is that we're sensing we've got fats and amino acids in the duodenum, right? So that fat was looping back and telling us to stop stomach secretion. But now on the pancreas side, that fat is telling the pancreas, hey, I need some lipases to help me break this down, right? And the same thing with the amino acids, right? Now that you have amino acids in the duodenum, you've got these protein products. You want to keep breaking them down. To do that, you need those proteases coming from the duodenum. Okay. So food in the form of fat and amino acids in the duodenum triggers the release of CCK, which tells our acinar cells to make our various pancreatic enzymes. Okay. For the secretin, its job is to make bicarbonate. Bicarbonate is a buffer, so it buffers out acidity. So the thing that makes sense and the thing that your body does in order to trigger the release of secretin, what you're going to be sensing is acidity in the duodenum, right? So if you've got acid in the duodenum, you need to buffer it out. So you need to make bicarbonate. The way to do that is to trigger this release of secretin. So we got pathways for this. So here we have our map, thinking about how are we neutralizing this acid. Okay. So we're seeing that we have acid coming into the duodenum, coming from that chyme, because we had the stomach making acid, right? We've got that stomach acid passing out of the stomach into the duodenum. In the duodenum, we've got some chemoreceptors, right? So receptors that are sensing chemical composition. In this case, what we're sensing is the presence of that acidity, the presence of those protons, right? Those hydrogen ions. So we're going to detect and respond. Some of these signals are kind of local. Some of them loop farther. So we have both short and long reflexes here. For our purposes, don't worry too much about the difference between short and long reflexes. If someone else tells you different in the future, come back and tell me and we can go into them in more detail. But we want to understand our general picture here. Okay, so we got these reflexes. They're telling your endocrine cells to make secretin. Then you've got secretin in your blood. That's what the endocrine system does. Hormones travel in your blood, travel in your plasma. Once we've got the secretin tells us to make bicarbonate. Specifically, we're making bicarbonate in those duct cells of the pancreas. We then pass them into the duodenum. So we're going out that ampulla of Vater and sphincter of Oddi into the duodenum. And that's where we neutralize the acid when we come into contact with it. Now, the fluid in our duodenum isn't so acidic, so it's not going to burn our intestines as it continues to pass through. Once the acid goes down, this whole signaling pathway turns off. So we got an example of negative feedback, because you only need this to happen if your duodenum is acidic. If it's not, then you don't have a problem. You don't have anything to fix. Okay. So it's not just pancreatic secretions that are coming into the duodenum. We've also got the bile, right? So we've got the bile salts, which we also have to control, right? So those bile and bile salts are really for the process of like emulsifying our fats. So we talked last time about how we have to break up those fat globules into smaller fat droplets so that the lipases from the pancreas can actually attack our triglycerides and break them up before we pull them into our lymphatic system as chylomicrons. Okay. So we got to do that. We got to tell our body to do that. And specifically, bile is coming from the liver. That's where we're making bile. All right. Let's use green because bile is green. Okay. So we got to make bile from the liver. And what's telling us to do that is secretin. So we've got the same kind of signaling coming, telling us that we have stuff in the duodenum. Since we've already got our signals telling us that there's stuff in the duodenum, we're going to use that same kind of signal to tell us to make bile. Okay. And the CCK now, instead of making enzymes, it's going to tell your gallbladder to contract. So your gallbladder is like a storage organ for the bile, right? So you made extra bile in the liver at some point, sitting around in your gallbladder tucked in there. When we sense that we have food and acid in the duodenum, that's sending signals to the pancreas to make enzymes and bicarbonate, sending signals to the liver to make our bile, and it's telling your gallbladder, okay, squeeze, give me that extra bile now. That's what the CCK is doing. And at the same time, we got a sphincter controlling all these ducts and stuff coming into the duodenum. Sphincters are circular, so we want them to relax so that we can actually get the fluids in there. So CCK is also doing. Okay, we'll stop there and pick up on whatever the day of the week is next. Okay. But we're about at the end of all this hormone stuff. We just got motility left, and then we'll break into reproductive either Friday or next week.